The overall goal of this experiment is to visualize in vivo blood vessel formation in three-dimensional scaffolds in real time by multiphoton microscopy.
This method can help answer key questions in bone tissue engineering field, such as how to visualize and track in vivo blood vessels formation in three-dimensional scaffolds in real time. The main advantage of this technique is that it enables to simultaneously visualize cells, extracellular matrix, and surrounding vascular networks in vivo.
To begin, prepare a homogeneous solution containing 20 grams of PLGA and 20 milliliters of 1,4-dioxane. Then, add 2 grams of powdered nHAP to the solution. Using a magnetic stirrer, mix the suspension vigorously at 1500 RPM at room temperature for 16 hours to form a uniform paste that will be used for printing porous PLGA nHAP scaffolds. Once printed, freeze-dry the scaffolds for 48 hours to completely remove any solvent, and then sterilize them in 75% ethanol for 1 hour. Afterward, lyophilize them once more to obtain neutral and aseptic material. Next, add 4.5 times 10 to the 5th of lentiviral particles to each scaffold and allow the particles to adsorb in a humidified incubator, maintaining 37 degrees Celsius for